Welcome to the Explainer. So, today we're exploring a concept that honestly sounds like pure science fiction, but it is rapidly becoming science fact. We're talking about building the world from the bottom up, literally, atom by atom. Just imagine for a second constructing entirely new materials, incredibly efficient computers, or even microscopic medical devices, not by, whittling down larger parts, but by snapping individual atoms together like the ultimate set of nanoscale building blocks. It's an absolutely wild vision, and it was primarily pioneered by Eric Drexler. As we go through this explainer, we're going to unpack the brilliant minds, the heated scientific feuds, and the stunning modern breakthroughs that are making Drexler's dream of molecular machines a reality. Here's our roadmap for today. We'll cover 1. The molecular assembler dream, 2. The Drexler-Sumley debate, 3. Wet chemistry versus machine phase, 4. Nature's nanotech blueprint, 5. Modern synthetic molecular machines, and 6. Policy, peril, and promise. Let's jump in. Section 1. The molecular assembler dream, from Feynman to Drexler. Now, to really understand where this entire field started, we have to look back to 1959. That's when the legendary physicist Richard Feynman asked a revolutionary question. What if we could manipulate individual atoms? That theoretical challenge just kind of sat there until it was enthusiastically picked up in the 1980s by Eric Drexler. Drexler effectively founded the conceptual basis of molecular nanotechnology by proposing something he called the molecular assembler. Think of it as a nanoscale desktop factory capable of manufacturing complex materials and devices atom by atom. He laid this all out for the public in his 1986 book, Engines of Creation, and then he followed it up with a deeply technical framework in his 1992 work, Nanosystems. The evolution of this technology actually spans decades. Following Feynman's initial spark in '59, Drexler published his landmark first paper in 1981, essentially arguing that since biological systems already exist, atom-by-atom construction is fundamentally possible. This paved the way for massive physical milestones like Fraser Stoddardt's 1991 invention of the rhodazine, which is basically a mechanical molecular ring threaded on an axle. And despite some incredibly fierce feasibility debates in the early 2000s, this foundational work ultimately culminated in a 2016 Nobel Prize for Molecular Machines. Section 2. The Drexler-Smalley Debate. The Clash of Titans. So while Drexler was out there championing these incredible engineering models and molecular assemblers, Nobel laureate chemist Richard Smalley pushed back. And I mean, he pushed back hard. In a 2001 article, Smalley argued that chemistry is an intricate dance of love between atoms, and you just can't force that dance with robotics. He raised two major objections, the fat fingers and the sticky fingers problems. He argued that any robotic nanoscale manipulator arm would itself have to be made of atoms, making it way too bulky or fat to control delicate chemical reactions. On top of that, because atoms naturally attract each other, these building blocks would stick to the robotic fingers, making it impossible to release them precisely. His ultimate conclusion, mechanical nanobots were simply impossible. Well, Drexler was definitely not about to let that stand. He and his colleagues fired right back, pointing out that his actual proposals never relied on the clunky manipulator arms Smalley was describing. Drexler straight up called it a straw man attack. He argued that Smalley was inventing a completely flawed mechanism, slapping Drexler's name on it, and then debunking it. Drexler insisted that his specific vision relied on rigid, mechanical positional control, which entirely bypassed the need for these impossible so-called Smalley fingers. Section 3. Wet chemistry versus machine phase. The core scientific disagreement. Beyond the whole fat fingers analogy, there was a deeply fundamental difference in how these two scientists viewed the future of chemical synthesis. Smalley was a champion of wet chemistry. He believed that complex reactions fundamentally require a fluid, dynamic liquid medium, specifically water, along with biological enzymes to properly orchestrate 3D structures. Drexler, on the other hand, approached this exactly like an engineer. He proposed machine phase chemistry. He argued for dry, vacuum environments, utilizing mechanical positional control to rigidly hold molecules in place. This would avoid unwanted side reactions and allow operations at incredible megahertz frequencies. Section 4. Nature's nanotech blueprint. How biology does it. So how do we bridge this gap? Well, we can look at a system that has been successfully running molecular machines for billions of years without any fat fingers getting in the way. Biology. Now, nature operates in a ridiculously challenging microscopic environment. At the nanoscale, water feels thick, kind of like honey. In physics, this is known as a low Reynolds number environment, where viscosity completely dominates over inertia. Furthermore,
Everything down there is constantly being violently shaken by random thermal energy called Brownian motion. Yet despite all this chaos, biological machines are unbelievably precise. Ribosomes act as true molecular assemblers, reading genetic code to build complex proteins atom by atom. ATP synthase spins like a magnificent rotary motor to generate cellular fuel. And motor proteins like myosin and kinesin act as literal cargo trains moving straight along linear tracks inside our cells. Biology proves, without a doubt, that molecular machinery is absolutely possible.
Section 5. Modern synthetic molecular machines. Human-made nanobots. The amazing thing is, we're no longer limited to just observing nature. Modern chemists are out there actually building synthetic molecular machines from the bottom up today, which is validating so many of Drexler's early theories. One astonishing technique is DNA origami. Basically, by taking a long single strand of DNA and using shorter staple strands, scientists can force the DNA to fold into highly specific, programmable 2D and 3D shapes. And because it relies on the natural self-assembly properties of DNA base pairing, it is massively parallel. A single operation in a tiny test tube can generate 1 trillion perfect copies. This gives us the ability to precisely position molecules and nanoparticles with nanometer resolution, which is exactly the kind of positional control Drexler dreamed of. And this synthetic approach allows us to build actual working motors, like a catenane motor. That's essentially a molecular system made of interlocked rings, kind of like links in a chain. In one recent synthetic system, a smaller, shuttling ring navigates around a larger track ring. By introducing a chemical fuel, the system creates what's called an information ratchet that literally forces the smaller ring to move in a preferred directed motion. The motor extracts free energy from the fuel to generate this directed movement. Guys, this isn't biology anymore. This is a fully autonomous artificial motor driven entirely by chemistry. Actually, building these incredible autonomous devices requires a perfect mashup of different scientific disciplines. To create a true molecular engine, we have to rely on physics to understand the complex thermodynamics and forces at the nanoscale. We need chemistry to physically build and synthesize these complex molecules from scratch. And we need biology to learn the fundamental design principles of how to operate efficiently in such a noisy, viscous environment. When these fields combine, the potential is literally limitless. Section 6. Policy, Peril, and Promise. The stakes of a molecular future. Now, if Drexler's ultimate vision of general-purpose molecular assemblers is fully realized, the stakes for humanity couldn't be higher. Looking objectively at the policy literature, we see profound peril on one side. Critics and policy analysts warn of the terrifying gray goo scenario, where self-replicating nanobots could theoretically mutate or be intentionally programmed to consume the entire biosphere. There are also severe geopolitical risks. Molecular manufacturing could lead to unprecedented, highly selective weapons that are incredibly easy to produce, potentially sparking massive, unstable arms races. But on the flip side, the promise is almost utopian. Proponents argue that desktop nanofactories could produce clean, wildly inexpensive, zero-waste goods. We could precisely target and cure diseases at the cellular level and even scrub the environment completely clean of pollutants. The disparity between these two possible outcomes makes policy planning incredibly, incredibly urgent. Which leaves us with one massive, ultimate question. Are we ready for a world built atom by atom? From Feynman's bold dream to the fiery Drexler-Smalley debates, right up to the DNA origami and synthetic motors being constructed in labs right now, the march of molecular machines is undeniably accelerating. Our physical capability to manipulate the nanoscale is expanding every single day. The science is finally catching up to the theory. The real test may no longer be whether we can build these tiny engines of creation, but whether our society and our policies are truly prepared for the immense power they're going to unleash. Think about that the next time you look at a drop of water or a grain of sand. There's plenty of room at the bottom, and we are just starting to move in.